Cauliflower is nothing but the cabbage with a college education. So that is said to be a cauliflower, a better vegetable than the cabbage. So, coming to the bacterial physiology, bacterial growth and multiplication. Growth is increase of all the chemical constituents or the components of the cell. Growth in bacteria occurs in two phases: increase in size or cell mass, then division of the cell into the daughter cell, that is increase in number. Whatever the balance between these two processes, net effect is increase in the total mass or the biomass of bacteria. Cell division is occurring by three ways. That number one, asexual by binary fission, sexual by conjugation, and the other's way of division, that is budding and fragmentation. So the first, asexual or the binary fission, here the single parent cell divides to form two daughter cells. Cell enlarge and single DNA molecule replicates, each copy attached to the cytoplasmic membrane. Invagination, that is the mesosome, cell begin to pull apart. Cell division occur by constrictive or pinching process in growth of the transfer septum across the cell, as one can push the finger in the balloon which is inflated. So this kind of septum occurs here, produce two cells of identical genetic composition. Daughter cell may remain partially attached after division in some species, so that is where the diplococci or diplobacilli or streptococci or streptobacilli are formed. Here one can see the biodivision, the plasma membrane, the cell wall, the DNA complete. It start swelling, the DNA duplicates, and half of this DNA that goes, and this constriction occurs, and that divides the cell into two equal halves, and half of the DNA is going into each of the daughter cell. Here one can see the constriction, the mesosome deepens and cut the cell into two parts. So the sexual cell division is conjugation. It's a type of the bacterial sex. Occurs only between closely related bacterial strains. Two organisms meet and share genetic information, such as a resistance to the penicillin. Donor bacteria grows tubes which are called sacs or F-pilars that project from its surface to the surface of the recipient bacteria. So the donor bacteria has got sex pilars which do attach to the recipient bacteria, and now a copy of the chromosomal DNA travels through this tube into the recipient, where it incorporates into the recipient genome. Here one can see the mobile plasmid, and that is having this sex pilars, and here it is the other recipient bacteria which do not have the sex pilars. So this donor bacteria which have the sex pilars do attach with this recipient, and they make a way by which this genetic material or this transposon or any gene that can be transferred from this bacteria to this bacteria, and then the new donor bacteria is produced, and these new donor bacteria are again able to reproduce by this way. So that is the bacterial conjugation, in which the different genetic material from one bacteria is being transferred to the another bacteria. The other modes of cell division, that is the budding. Bacteria like Mycobacteria, Corynebacterium and Hypomicrobium divide by the budding. The budding cell originates as an outgrowth of the original or the mother cell, which eventually reach to the size of the mother cell. The bud now separates to have independent existence. It is just like as occurs in the yeast. So this budding also occurs in the bacteria. Then the fragmentation. Several actinomycetes form filamentous cells that reproduce by simple fragmentation of the original filament into the new cells. So that is called fragmentation. Here one can see the budding nucleus replicates and the new nuclei moves to the bud that originates as the outgrowth from the mother cell, and this young bud do continue to increase in size. It receives the genetic material from the mother cell and ultimately it gets detached from the mother cell and become the daughter cell which can live independently. Here the type of fragmentation, as one can see, the end walls grow inwards, the ingrowth enlarges and ultimately the filaments breaks and the fragment separates to exist as the independent cell. So this is called fragmentation. And under ideal condition, the bacteria grow very rapidly. Some double in number every 20 minutes. That is called doubling time. Doubling in number occurs like that: 1 into 2, 2 into 4, 4 into 8, and 8 into 16 is the exponential growth. It starts off slowly, but once going, number of bacteria increase very rapidly. Usually some nutrient runs short or the waste material builds inside and the growth ceases. Eventually a die-off occurs, reducing numbers of the live bacteria. All it is given in the bacterial growth curve. And here it is the bacterial growth curve. When the bacteria is grown on a suitable liquid medium, its growth follows a definite pattern or the course. Bacterial growth is exponential only when conditions are ideal. Rate of the growth is modified by the different environmental factors. Samples are taken at the regular interval. Bacterial counts are done and plotted in relation to the time which yield a characteristic growth curve. Changes of the slope on the graph indicate transition from one phase to another. Growth curve of the actual population shows 4 distinct phases. And these are: number 1, lag phase or the slow phase; log phase or the rapid or exponential or logarithmic phase; stationary phase or the plateau; death phase or the decline phase. Various stages of the growth curve show morphological and physiological changes of the cells called as cell changes. So here one can see the growth curve, log number of living cells or the turbidity that is plotted on this side, and the time usually in hours is plotted on this side, and this is the growth curve. That is the 4 parts of the growth curve: lag, log, stationary and the death. Here is a really beautiful painting Sanna made, and the nice thing about it is that the painter have made the legs of the horse, giving the impression of the eyes. So the lag phase or the slow phase. When the bacteria are put into fresh culture medium, no immediate increase in cell number occurs. There is increase in the cell size first to get the maximum, but little or no cell division occurs. Here bacteria are adapting to new environment and nutrition in media. All there is little or no growth, no cell division, so it is called lag phase. Bacteria are not dormant but collecting the necessary and the new enzyme and the metabolites, preparing for the cell division in adequate quantities. These things are collected to start multiplication. Length of the lag phase varies with the species, nature of medium, and environmental factors. Here one can see the lag phase, here the viable cells, and here are the hours. So one can see green color of this tube indicating few or no cells. So this is the lag phase. The next is log phase or rapid or exponential or logarithmic phase. Here cells start dividing, their number increases exponentially 1 into 2, 2 into 4, and 4 into 8. Growth rate in cell division is maximum fast, so that's why it is called logarithmic. So graph is a straight vertical line. This phase is used to determine generation time, or in other words, doubling time of the bacteria. Here nutrients are sufficient, positive balance, and little competition. Phase is of short duration, so ends because of the exhaustion of the nutrients in the medium, which has been used very rapidly by the bacteria to divide and multiply, producing more. So accumulation of the toxic metabolites and products, rise in the cell density, population bump, and change in the pH, decrease in the oxygen tension for the aerobic organism. In log phase, cells are most active, used for the industrial purpose, but bacteria are very sensitive to radiation and antibiotic like penicillin and others. These interfere with the growth process. So these antimicrobials are most harmful to bacterial cell during this phase. Cell changes are, the cells are smaller and stain uniformly. So here one can see the exponential or growth phase, the tube showing the blue color, and the blue color is consisting most of the live cells, and few dead cells start appearing, that is the red, which is showing the dead cell. Now the third phase is the stationary phase or the plateau phase. Here cell division stops due to the depletion of the nutrients and accumulation of the toxic products. Bacteria stop growing. Total bacterial cell number reaches the maximum number. Number of the progeny cells formed equal to the number of the cells that do die. So equivalence means the equal number of the cells are produced and equal number of cells are dying. Viable count remains stationary because of this equilibrium. Graph shows a plateau or a horizontal line, and the cell changes are gram variable with irregular staining. Some bacteria undergo sporulation and alarmones is induced. These are said to be the chemical substances which are responsible for stopping the growth of the bacteria. So here one can see the stationary phase, and if you look at the tube, it is having the live cell as well as it is having the dead cell, but these are usually of equal number. So the net result is the equivalence. Now the last is the death phase or decline phase. Rate of the death increases, rate of the reproduction viable bacteria decreases. As nutrient supply is exhausted, bacteria start to die, so that's why it is called death phase. Like bacterial growth, death is exponential, cell death, nutrients consumed, so starvation like condition occurs. There occur bacterial lysis with build-up of the toxic waste and autolytic enzymes. Growth rate becomes negative, graphs start falling, and the cell changes are involution forms are common, that is smaller, or here one can see the death phase, and here the dead cells are more and the live cells, the blue color, are less. Now the summary of the phase of the growth curve: lag, the adaptation to the nutrients; log, the active growth producing very rapidly the number of cells, stationary the death equal to the growth rate, and the death nutrients consumed, pH too low, and ultimately the cells start dying. Here is a beautiful painting, and one can appreciate the color of the water of the sea that is coming out from the painting the painter have made. It's imagination, and really very nice and creative imagination. Now coming to the bacterial count. It is the measuring the bacterial mass, live and dead. Growth of the bacteria is diffused in liquid medium but form colonies on the solid medium if are grown on the petri dishes. Each colony consists of the clone of the cells derived from the single parent cell. Bacteria are counted by two methods. Number one, total count, and the number two, viable count. So the total count, total number of bacteria present in the specimen, whether living or dead, bacteria are counted under microscope using counting chamber or by automated machine. Then viable count, to measure only the viable living cells, not dead cells. These are capable of growing and multiplication. These produce a colony on a suitable medium, and determination of the viable count is done by dilution method or by plating method. Here one can see the chamber by which we can able to count the bacteria, and this is the machine automated which can count the number of bacteria seen in medium. So bacterial count is done by dilution method. Suspension is diluted to a point where unit quantities do not grow when inoculated to the suitable liquid medium. So no growth occurs on putting this to the medium. It does not give accurate values, so used in coliform count in the drinking water. The other method is plating method. As the name is, here proper dilutions are inoculated on solid media by pour plate method, and the other is spread plate method. Serial dilutions are spread on the surface of the dried plates, and the colony counts are made which do appear on that dishes. Here serial dilution, one can see that we put this material here, and we take 1 ml of this and put it to the other already formed this tube, and 1 ml from this inoculated material to here, then from this 1 ml to the another this to make dilute, and then from this 1 ml to this tube, and from this tube to this tube. Ultimately, when that tube shows no growth, here one can see the maximum growth, lessening of the growth, lessening, and ultimately no growth here. And here is the spread plate method. Simple 0.1 ml of the liquid material is poured onto the solid medium and spread simple over the surface, and then the plate inoculated and bacterial colonies are counted, and this is called spread plate method. That is the end of the lecture, and I hope you are going to subscribe it and you will like it, and do share it to others if you like it. And if you don't like it, you have the right to have any kind of question from me. My numbers are already given, and I am thank you very much for having this video, seeing and enjoying. Thank you.